Okay. So for our November seminar, I want to welcome Dr. Sara Gushgari-Doyle from Berkeley National Labs. She's a volunteer who, Jaylene reached out on Twitter and said, we want someone who's going to sort of span the gamut of all the interests in our department and also education, and I think she does it well. So her background is actually in civil and environmental engineering, and from that she got involved with bioremediation, got further involved with exploring how different bacterial and microbial metabolic pathways might be explored and applied, and really into the genomics of microbial metabolism, but also still continue to pursue her interests in education and STEM outreach, particularly at the K through 12 level. So I think she'll be of great interest to everyone in this audience, and look forward to hearing more about her research myself. And with that, I'll turn it over to her.
Thank you so much for that introduction. Let me share my screen to start with. All right, so I'm Sara Gushgari-Doyle, and I am a postdoctoral scholar here at Lawrence Berkeley National Lab in Berkeley, California. This is actually one of the views from our lab behind me in my little Zoom background. And today, I really want to share a story with you about connecting genotype and phenotype to ecotype, and the environment that some of these organisms are living in and come from. And we're specifically going to be talking about organisms in the terrestrial subsurface. And when we're talking about this and discussing this, I want you to sort of think about all of this in the lens of competition in an environment, or an organism's strategy to survive. Now, before I get into the science, I always like to start with acknowledgements and thanking my collaborators and colleagues. The work that I'm going to be sharing with you today is funded by a large Department of Energy science focus area called ENIGMA. And ENIGMA is a massive team science project. We have 17 or 18 principal investigators across 13 institutions. It's, a huge team. And because of that, we can really get at a systems biology approach and understanding of what is going on in the ecosystem that we study. We have bioinformatics specialists, we have molecular biologists, hydrologists, all of these people coming together to really understand the system. And so these are just a few of my colleagues I wanted to highlight specifically for this work. My principal investigator, Romy Chakraborty, Professor Adam Arkin down at UC Berkeley, and Professors Terry Hazen, Matthew Fields, and Mike Adams and their teams really contributed a lot to this work. And then I also have my contact information down there in the lower right, email, and on Twitter, as was mentioned, how I found you guys. And I'm always totally open to chatting about science or outreach or anything, so feel free to reach out. Now, to begin the story, I'd like to set a frame of reference and give some background into our field site and to how these organisms are living out in the environment. And so our field site is the Oak Ridge Reservation Field Research Center, or the FRC, the ORR-FRC. I'll use all of those sort of interchangeably. It's all the same site. And it's located out there in eastern Tennessee, as you can see through these maps in the sort of circled region. And the map on the left shows both uranium and nitrate contamination in the United States. Uranium is those yellow to red color scale, and then the blue color scale is the nitrate. And we see a lot of nitrate contamination in agricultural zones, as you would expect, right? Everybody hears about the nitrate and nitrogen contamination associated with synthetic fertilizers. And so a lot of times it's centralized in agricultural zones, like in the sort of central United States here. Let me change this to a pointer. Sweet. Here in the central United States, and then over sort of by me, in the Central Valley, California, of course, has a ton of nitrate contamination. But our site at the FRC is not agricultural related. And so why do we have that nitrate? So this other map that you see on the right is a Superfund contaminated site map put forth by the EPA. And that's, of course, then why we're here at Oak Ridge studying this site, because of this heavily contaminated environment. And at Oak Ridge, we have this high uranium and nitrate contamination because Oak Ridge was one of the sites that was involved in the Manhattan Project back in the 1940s. And they were specifically looking at processing of uranium there. And then, of course, because of that, as all industrial processes go, they made a lot of waste. And as you can imagine, back in the 40s, 50s, 60s, 70s, even beyond, we did not have a ton of engineered controls in place for environmental protection. And so you can see in this picture on the right here, these waste disposal ponds that were just sort of open to the air and not very well lined. And that's how they were disposing of their uranyl nitrate waste at the site. And so eventually the EPA steps in and says, hey, this is a bad idea. We need to fix this. We need to do something about this. And so they capped it. Well, first they neutralized the waste because it was very, very acidic. And then they did a seal and a cap over it as a remediation strategy. You might think that sounds really silly as a remediation, just to put some concrete over it, but actually that's a lot of times how remediation goes in some of these sites. And so now we have a parking lot that looks suspiciously like those ponds over there, and it's considered, remediated. And we still have a ton of groundwater wells that we monitor here for our study. And so this really contaminated waste has impacted our environment and the surrounding ecosystem massively. And so we have groundwater wells that we sample. We sample sediment cores pretty frequently, and still we observe pHs as low as 2, sometimes even a little bit lower in the surrounding area. And then we had a sediment sampling campaign recently this year that we measured up to 19,000 ppm of nitrate in some of these soils. 19,000 ppm. That is a ton of nitrate. And it's even more sort of perplexing and puzzling because nitrate, as I'm sure a lot of you have studied before, is one of the primary commonly used terminal electron acceptors. So how is it just hanging out here? Why, we observe a lot of microbial activity through different measurements. Why is all this nitrate just hanging around? And so that's sort of the impetus for the general project, and to understand this weird ecosystem in Oak Ridge, Tennessee. And another interesting part of this site has to do with the actual structure of the subsurface. And so this plot that you see here has elevation below ground surface in meters here on the y-axis, and a horizontal distance of meters on the x-axis. We have the water table shown here by that caret in line, and then all of these vertical sort of cylindrical pipe-like images there are where we took those sediment cores that I was talking about this year. And all of the different colors are different sediment and soil types. And so, we're categorizing things in the realm of clays and silty loam and all of those things, but even not knowing which each of the colors represent specifically, what you notice here is that this subsurface is incredibly heterogeneous. And so that really creates interesting flow paths of the groundwater that's flowing through. And so, what this all sort of results in are lots of very different microniches in the terrestrial subsurface. You have sites or cores right close to each other that seem to have very different geochemical properties. And so that is really going to influence our microbial communities and how they're structured and how they function. And so when we characterize these sites, we look at tons of different measurements. As I said before, we have a very diverse team, which is able to take these excellent measurements. We, of course, characterize things like moisture and porosity, lots of metals. We've done some pretty substantial work in molybdenum and tungsten. We're able to measure activity through technologies like BondCat and gene classes with metagenomics and geochip and things like that. And we're able to study all of these parameters, not only spatially at the site, but also with depth. And so for this specific story that we're going to be talking about, we're going to focus in on a single well. So the samples that I'll talk about from here forth were taken from a single planar location, and it was a sediment core that we took with depth. And so we were looking at sort of what I call the terrestrial subsurface. It's sort of below the soil horizon that is the superficial soils that people have studied in great depth. And we're looking at about 1 meter below the ground surface down to about 5 meters. So it's also not the deep subsurface, right? It's sort of this in-between zone. And we're looking at sediment there from those depths of 1 meter to 5 meters down. And then also the groundwater that came out of the adjacent well just about a meter away. And so from all of these different depths and the groundwater, we were studying the microbial communities and we performed a bunch of isolations. And from every single depth and the groundwater, we recovered this Arthropacter. And Arthropacter is known as an environmental organism to take a lot or perform a lot of complex carbon transformations. So, big molecules like different carbohydrates and things like this. They're really known for those sorts of metabolisms. And when we sequenced these Arthropacter and, did a phylogenetic tree, this is just showing a phylogenetic tree of the full 16S sequence, we saw that they spanned, a pretty high diversity of this genus. And so this tree here is rooted up at the top with a closely related genus. And then all of the other Arthropacter that are included, sorry, let me back up. The Arthropacter from this study are highlighted in the colors that correspond to their little dots here on that figure. And then all of the other Arthropacter that we included are also environmental isolates that were publicly available or published previously that had complete genome sequences, so fully circularized genomes. And we selected those because we also fully circularized the genomes for this study. And so for further genomic analysis that I'll talk about in a little bit, we wanted, really comparable genomes to look at. And so, we cover a pretty large diversity of these Arthropacter, and some of them are pretty closely related, like you'll see. There's a group up here that is closely related, but none of them are actually the exact same strain. And so to start looking at these Arthropacter and we want to see why different Arthropacter that are different strains and supposedly have different functions are located in all of these different zones. And so we tried to first connect phenotype to ecotype and see if we could connect some of these geochemical parameters from the actual sediment and groundwater samples themselves to laboratory assayed phenotypes of these Arthropacter. And so we looked at some parameters like pH and chlorine for salinity and several metals, and I've only highlighted a handful here. Of course, we looked at that whole panel that I had talked about earlier of all these different, geochemical parameters that we measure. But when we looked at then the phenotyping, we saw some connections of different organisms sort of correlating to their environment of origin. For example, we had the highest tolerance for salt observed in that very deep sediment saturated zone Arthropactor, in which we observed the highest salinity. And then the IC50, or the concentration of salts that caused a 50% decrease in exponential growth, was 7% salt for that isolate. Similarly, we looked at pH, optimal pH growth ranges. And, through the entire sediment column, we actually had pretty consistent pHs, even in the saturated zone down here. And I should have mentioned before that the CF here in this figure stands for capillary fringe, or the sort of variable saturated zone in the terrestrial sediment, and then of course the vadose zone above that. And so we had pretty consistent pH down the sediment column, but the groundwater pH was closer to 8, as opposed to the roughly 5.5 pH of the sediment. And again, similarly, we found that the highest optimal pH was found in groundwater isolate 123 up here. A couple other things that you might notice in these graphs is that this isolate here, and then the one, oh, I can't drag because the bars are in the way, that's okay. And then the one to the right of it are two groundwater isolates, and they're very different. And that's actually a common theme in this study, is that the results of phenotyping and even the genomic analysis of these groundwater isolates are so different. And that sort of reflects this idea that groundwater in these environments is so transient. And because of that, these organisms could have come from anywhere. We don't really know what the actual environment of origin is for these groundwater organisms, since they tend to travel with the groundwater. But that's sort of an aside. And another interesting result of these pH analyses is that there is a really wide pH range for the sediment organisms, which is everything else below, for which they had pretty comparable exponential growth rates. And generally in bacterial microbiology, that's something that we don't see as much. We generally see really nice, pretty, curves with an obvious maxima and a really narrow range that the bacteria are happy and thriving in. But this sort of wider range is more characteristic of, some higher and eukaryotic organisms. And I think it might be a function of just that we haven't, explored all of these other ecosystems as much as some, more well-characterized ones like the rhizosphere, which has been, very heavily studied, and surficial soils and things like that. And so there's a lot of really cool things we found from this study, but I could, digress for days. Going back to trying to tie the phenotype to the ecotype, we then looked at some enzymes, especially complex carbon transforming enzymes that involve manganese, which a lot of these enzymes are dependent on manganese as a cofactor or something like that. And so specifically, what I'm highlighting is the manganese dependent peroxidase. And we did find that these deeper sediment isolates, which represent, these 3 dots right here, had these manganese dependent peroxidases. And then, of course, our groundwater outlier that I mentioned, that the groundwater results were all over the board. And, we also had our higher concentrations of manganese observed in the deeper depths. And then similarly with iron, we observed the highest number of siderophore biosynthesis and transport genes in the groundwater isolates. And of course, that sort of mirrors the lower bioavailability of iron in the groundwater as compared to sediments. We did try to test this one in the laboratory using a chrome overlay assay for siderophore biosynthesis. Unfortunately, this particular assay did not give positive results for our groundwater isolates. But of course, this could be a function of, there's a metabolic precursor that we don't know about that these guys need to produce the siderophores, or perhaps there were a different structure of siderophore that this doesn't account for. And so we're continuing to look for other siderophore assays to expand our repertoire there. But in addition to these sort of geochemical connections to, pretty standard phenotyping for isolates and environmental isolates specifically, we also wanted to tap into that story of this complex carbon transformation and what the differences among the isolates might be there. And so we looked at both the genomes and then also some phenotypic assays of complex carbon transformation by the Arthropactor. And so here, let's start actually on the upper right, we have the numbers of different carbohydrate active enzymes in each of these isolates, separated by the groundwater guys versus the sediment. And right away, you don't see a huge, obvious correlation of numbers of total enzymes, but we do see that they all have a lot of this GH in blue, which is the glycoside hydrolases. And so we started from there and said, okay, well, let's just test some known glycoside hydrolases to see what the activities look like. Are they any different than, just having the gene? And the answer is yes and no. And so, for example, with the genes such as, or coding for enzymes like the beta-glucosidase, they all had pretty high activity. But when you look at the results from this xylanase assay, we have these deeper sediment isolates, again, having really high xylanase activities as compared to the groundwater and more shallow sediment isolates, which demonstrated significantly less activity. And just to sort of give a frame of reference of what these assays are and what they're measuring, we have OD590 on the y-axis, which of course is commonly used for just cell density, but that's not what these are. We utilize some assays that use color tagged substrates. And so we have, for example, cellulose or xylan or hemicelluloses and things like that, that are actually tagged with a blue dye. And so when the organism utilizes this enzyme to cleave that bond, it releases the blue dye into the supernatant and into the liquid phase. And so then you can spin all the cells out in all of these solid substrate, because a lot of these are not soluble, and then measure the color of the supernatant just in your regular plate reader. And so that's what these assays are. And so we have these differential activities and things like that. And so how does this compare to what the carbon actually looks like in the subsurface? And so we were able to look at the different fractions and relative abundance of these fractions of some different complex carbon types in these sediment samples. And we did this using a mass spectrometry technique, FTICR-MS, for your, oh gosh, transform and something or other. I do not know what that is off the top of my head, but basically it's able to give us these sort of qualitative analyses of what the different compounds might be. And so we don't have, an absolute of this quantifiable number of this specific carbohydrate, but we do have relative abundances of, what types, general types of carbon are there. And so what we were able to see from this is that specifically these carbohydrate esterases, which is the green bars here, sort of correlate with the amount of carbohydrates that are present in each of these depths and then in the groundwater. And unfortunately, with our smaller sample size, right, we only have 7 isolates here that we're looking at. It's not a statistically significant correlation, but they definitely trend together. And this sort of finding and discussion led us to want to look at a greater number of isolates, right? Like, let's see if these trends that we're seeing in the genotype and phenotype to ecotype extend beyond our one sampling site in our one location. And so to do that, we come back to the phylogenetic tree that I showed before. And again, we compared all the Arthropacter that are publicly available with complete genomes, and including ours, this is 41 different genomes in Arthropacter. And they come from different environments. Unfortunately, we have a pretty big unknown group, because historically we as scientists have not been the best at cataloging our metadata. But we also have surficial soils, we have mine soil and desert soils, fresh water of different types, some permafrost, and then one wastewater that we included in there. And they also came from different countries around the globe, different continents. And so we have a pretty decent set and diversity of these Arthropacter, especially considering it's a less studied genus, and just in general, the amount of isolates that have come from environmental sources versus clinical sources is very, very different. But to start with, we first looked at just the amount of carbohydrate active enzymes in all of these different isolates. And so first here on the left, we have our total number of CASI enzymes, the carbohydrate active enzymes. And then we also broke it down through the different sort of categories, including those carbohydrate esterases, the glycosyl transferases, and things like that. And we did also run statistical analyses on these using just ANOVA statistical analysis. And these two, the carbohydrate esterases and the glycosyl transferases, did show significant differences among the groups, right? And so in the glycosyl transferases down here, it's pretty notable that the permafrost organisms have significantly lower numbers of glycosyl transferases, whereas over with the carbohydrate esterases, we see those significant differences in the number of CE enzymes that the endosphere isolates had, and also our terrestrial sediment guys. And in addition to looking at just the carbohydrate active enzymes, we wanted to compare the genomes on a whole, right? Compare these organisms in their entirety. And so that's where we came to this pan-genome analysis. And if you haven't seen a pan-genome analysis before, or even if you have, it's great to sort of refresh yourself on what these mean and how to read them. And so we have this massive circular plot here, right? And each of these lines is a genome in itself. And so we have all 41 going out. And they're ordered in this plot as the number of genomes they appear in. So over here on the right sort of horizontal area, we have the genes that appear in every single genome, all 41. And then moving, clockwise, we then have we call that the core. And then moving clockwise, we have the soft core, which is a very small group, and it is representing gene clusters that are present in 95 to 99% of the genomes that were analyzed. And then over here, jumping to the cloud, which is this massive part of the plot on the left, these are gene clusters that were only found in 1 to 2 of the genomes, and that's so 1 to 5% of all the genomes analyzed. And so in our sample size of 41, that's 1 or 2. And then the shell over here is the rest. And so we looked at all these gene clusters, and one way of analyzing them is we sort of grouped them by KEGG module, right? So functional modules. And what we saw is that the core and soft core, which we frequently sort of group together to sort of account for maybe shifts in open reading frames or weird annotation. So we sort of group core and soft core as gene clusters that everyone has. And a lot of those were, first of all, your single copy genes that we look for in, these analyses. They were general housekeeping genes, your DNA repair, your ribosomes, your tRNA biogenesis, things that we consider, essential to life. And then the shell and cloud was a lot of genes that are what I like to call sort of for adaptation to your environment. They were, carbon transformation enzymes. I plotted specifically the carbohydrate active enzymes again here in panel B. And, the core has a few because Arthropactor are known for, this complex carbon transformation metabolism, but largely they are in the shell and in the cloud, and they're in great many numbers, right? We had 300 and some and almost 450 carbohydrate active enzymes in the shell and the cloud of this pangenome. And they also, let's see, some of the, where am I? That's okay. Some of the shell and cloud genes were two component systems was a big category, a lot of transporters and things like that. And so, we have all of these genes that might, be specific to a certain environment or a certain toxin in the environment and things like that. And so in addition to just looking at the genes, we can quantify a pangenome using this simple little equation here, G equals C n to the power of gamma. And where G is the pangenome size, so all of the genes in this entire sort of visualization of gene clusters, excuse me, we have the core genome size, which would just be these guys in green over here. The number of genomes, ours is 41, and then this openness coefficient, which is what we're solving for. And the openness coefficient ranges from 0 to 1. And when it's close to 0, we say that it's a closed pangenome, which means that a lot of the genes in the pangenome are in the core and the soft core. So they're shared among each other. And then when it's very close to 1, it's the opposite that is true, right? We have tons of genes in the cloud and in the shell. And generally speaking, in a lot of the studies that I've seen of just one genus, the gamma values are much lower than our gamma value, which is 0.9. It's very close to 1, right? And in some of these other analyses, we're seeing gamma values closer to 0.1. Or sometimes up to 0.3 and things like that, but 0.9 is somewhat uncommon for a pangenome of one genus. And so in addition to looking at a pangenome, we can also think about other markers of sort of this lifestyle of being able to adapt, right? And sorry, before I go on, I should just also say that with other folks who have been studying the this adaptation of microorganisms to different environments or to a perturbation or things like that, they have identified this open genome as a signature of that. And that's, sort of what we're finding here. We have these, different environments, especially with the Arthropactor that we studied here. They're from, we had permafrost and glacial freshwater, the terrestrial subsurface, the soil. They're very, very different environments, and we're seeing, very, very different genomes. And so in addition to this pangenome approach, we can also look for, another classic signal of adaptation, like mobile elements. And so in this study, we then, just going back to our 7 isolates in these figures, we looked at genomic islands and the genes that were around those and associated with the genomic islands. And then we also looked at plasmids. And in these organisms, only 2 of them did not have plasmids. One of the organisms had 3 plasmids, which is a fun facet of being able to fully circularize these genomes. You also get these fully circularized plasmids along with them. And some of the classic plasmid genes that we think about are things like antibiotic resistance, right? Different toxins and things like that. But what we found in these sediment and groundwater plasmids or microorganisms plasmids was not really along the lines of what we typically think. We found carbon metabolism. specifically also metals resistance and different transporters. Some of the same genes that we were finding in the shell and the cloud of the pangenomes. And then it was similar with the genomic islands. Around some of these transposable elements, we were finding, other genes that are, more associated with an organism trying to live and survive in its surroundings. And we loved this result, right? It was very exciting to see that, some of these environmental genes were around these signatures, classic signatures of adaptation. And so in adaptation, and another phrase that we sort of toss around, genomic plasticity, just to talk about historically what we think about that again is that virulence, pathogenicity, antibiotic resistance, this broad, vague catabolism, which our carbon transformation metabolisms do fall under that, but I think that was sort of just a throwaway bucket of terms that people used to use to talk about genomic plasticity and adaptation. And I think a lot of this is, again, because of this historic focus on studying microbiology in the lens of clinical studies and of medicine. And so what we can add to this now is environmental stress and variability, right? We have a high variability in the different environments that all these Arthropacter and the pangenome came from. We have, in my smaller study of the 7, we had weird contamination conditions that were sometimes low pH or sometimes higher nitrate and things like that, and metals contamination. So sort of stressful conditions that an organism might need to adapt to. And so then if you consider an organism like Arthropacter that has these plasmids, has a bunch of these mobile elements, has a sort of open pangenome across the genus, you wonder, is this a way that they are actually coping with this need to adapt and acclimate to their environments? And I like the term genomic strategist here, because in classic competition in microbiology, we talk about K versus R strategists. Are these organisms using their speed of metabolism to be able to outcompete? Are they using their enzymes that have a very, very high affinity for the substrate to be able to live in these, lower concentrations of their substrate conditions? And here, do we have genomic strategists who can shift around their genomes to see what they need? And so, there's a lot of questions to answer before we can say much about this idea of genomic strategy. So where do we go from here? Of course, we need to do analyses of more comparable pangenomes, because pangenome analysis and pangenomics is a relatively new sort of approach and field of study, right? And so we don't really have hard and fast rules for what is the acceptable protocol and standards and the way that we look at these pangenomes, right? So I was talking about how we used all fully circularized genomes, and that was a choice that we made for what we wanted to be able to look at. And so we need to be able to sort of compare across these pangenome studies to be able to say more about pangenomics. And then, of course, analysis of accessory genomes across more environments. And I didn't use this term before, and that's my fault, but the accessory genome being those shell and cloud genes, the ones that are not in the core, would be the accessory genome. And so are we seeing that we have a similar comparison of accessory genomes if all of our samples are as diverse as this Arthropacter study was in their environment of origin? more whole community genotype to ecotype, because as fun and interesting as these isolate studies are, these organisms are not living alone out in their environment, right? They have an entire community that they're interacting with and that, they're competing with and things like that. And so, of course, you got to start somewhere, but perhaps like a bottom-up approach to this sort of whole community analysis, moving to more complex consortia, eventually to whole enrichments and communities. And then the question also comes up from this analysis of how do we categorize taxa? Because it could be argued that we see this massive open pangenome because the Arthropactor really should be several genera, right? And that's a sort of larger question in ecology, and especially in microbial ecology, of how we categorize the different taxonomic identifications. And, that is a whole other ball game that is going to need tons more research and discourse within the scientific community, because it's a big question. It's an important question. And so I just wanted to thank again my whole team of collaborators for all of their input into this work. And I also just quickly wanted to share some about my outreach, which I have a few slides here at the end. So if we don't mind, I can quick talk about this also before we open the floor to more discussion. And so as was mentioned before, I am, I hate to use the cliche word passionate, but I'm very passionate about education, about STEM outreach, and about specifically, bringing equity and inclusion and everything more into STEM. And I think a lot of this happens at the level before college, right? To attract and retain more diversity, we need to start before we even get to college. And so a colleague of mine here at Lawrence Berkeley and I co-founded this nonprofit organization called STEMC. And that stands for Scientists, Technologists, Engineers, and Mathematicians Sharing Ideas. And the idea of this project, I guess, is to tap into our network of scientists, of experts, and of engineers in industry, and all of these people who have the up-to-date knowledge of what's going on in their field. And we're using our network of these experts to make videos about different topics for different age groups and things like that. And we've also recently started to do articles as well that could be used in curricula for, not so much K through 12, but for middle and high school. And, so why STEM equity? I'm sure, a lot of you think about this a lot and have been involved in these conversations, but the National Science Foundation puts out this study of sort of the demographics of STEM fields every few years. And so in the last one, which was published in 2019, they found that 72% of American STEM PhD graduates were white, 72%. And then, similar metrics of Americans in STEM careers, only 7% and 9% were Black or Hispanic Americans, respectively. And this is not okay. This is, not the way to produce good results, scientific, engineering or otherwise. Studies have shown that diverse teams, make fewer mistakes and more profits, and for even just looking at this in the realm and the lens of productivity. And so we are taking, again, as I mentioned, our broad network of STEM experts to develop this content, because they are the real experts, and distributing it open access entirely on social media and YouTube and things like that for students to use, for educators to use. And of course, what we saw in the past 2 years or so is that translatable learning tools that can be in the classroom or also be used remotely and online are more important than ever. And so we have started putting out a few videos with our collaborators. You can go to stemc.org to look some of them up. They range from sort of a basic introductory science ideas like the scientific method to some of our research colleagues who have been, doing flash talks about their research and about really cool aspects of what they do. So one that was recently posted was a colleague of mine who works in creating algorithms to manage like internet networks and try to avoid congestion and traffic in the internet, which is the last I can say about that, because I have no idea what else this field is about. It's of course a very different science than what I do, but just wanted to sort of talk about this a little bit. And we're always looking for people to contribute content, articles and things like that. And so we're also always, trying to get the word out there to educators that we have this resource here that's completely open access and free. So yeah, that's STEMC. But with that, thank you all for inviting me and for your time and attention. And I'd love to discuss, anything we talked about today. So thank you.
Thank you very much for sharing both that immense amount of work and for your work with STEM education. I'm definitely going to go check out the STEMC website. If anyone has any questions, you're welcome to either speak up or raise your hand.
Should I stop share for now so we can go back to sort of a discussion screen? Or
yeah.
Can I ask a question?
Yeah, absolutely.
So, okay, I want to make sure I got this right. So you said that the higher, like in the areas that we looked at, there was a really high diversity of genome. Is that right? Or like there's a larger genome?
Yeah, so you're closer with the diversity of genome.
Okay.
So what we were seeing is the genomes of even the isolates from that same location were very, very different, but they're really closely related with 16S. And so, we think about categorizing these organisms by 16S because they're supposed to be closely related, right? And they're supposed to sort of manifest the same functions and things like that. But with this study, we saw that their genomes and their, potential metabolisms were very, very different. So it has like a very diverse genomes. So exactly what you said there.
Awesome. Thank you so much.
So I have a couple of questions. One of them is just how general do you think this result is? This kind of openness of the genome in these in bacterial species. Is this something that's maybe more unique to your site, or something that's just more general? And the second question is, for a student that might be interested in going into doing this type of research, looking at genomics and understanding functional, maybe biochemical diversity as well. What are the types of things that they would need to do to develop the skills necessary to do this type of research?
Great. So I'll answer that first question first of how generalizable do I think this is. And so I do not think that it's specific to our site. I do think that there's probably a higher diversity within genera that are more associated with environmental samples, which of course is more than we think, right? We see a lot of taxa lately that have been previously associated with hosts, be they human or whomever, that we are now seeing in environmental samples. And it's really just a function of we're starting to do a lot more environmental research. And so I think that it's more a function of these genera that seem to be found everywhere, for lack of a more specific way to put that. These ones that can thrive and survive, even if they're not, maybe the biggest member of the community, that they're still active and living in a wide variety of environments and conditions. And then to answer your question about sort of what students can do to get into this sort of research, I think moving forward, especially in biology and ecology and things like that, I think coding skills are so important moving forward, and not always something that's included in your curriculum. It might be in some of the in different departments, but it's less universal than I think it should be at this point. And so, just even taking a handful of coding classes, learning some of the basic languages like Python and R, and, if you're really into it, C++ and things like that. But I think that's really important. I think also just, developing your critical thinking and how you approach questions is important in any research, regardless of what field you want to be in. And so, be critical of things, ask a lot of questions when you read things for class, when you consume, your Twitter feed or your TikTok feed, be critical and thinking of like, is this, I guess less for TikTok, maybe Twitter and news and stuff. Is this bogus? Like, what are some of the biases that might be at play here? What are some of the, things that they might not have considered? I think that's a really just important frame of mind to approach anything with when you're a scientist.
I think Sara had a question.
Yes, can you hear me?
Yes.
Okay, so I have a question about what you were saying almost at the end of the presentation on regards of how we tell taxa from each other. I know it's difficult being microbes, right? It got me wondering how do you name taxa with microorganisms in that way? I'm just curious.
Yeah, absolutely. So right now, and historically, the standard practice for naming taxa and for delineating taxa in bacteria and archaea has been specific cutoffs of identity among 16S sequences. And that's it. And so we're looking, at how similar are these sequences, but and 16S is a good proxy, right? You can find it in, nearly every single thing we've looked at in bacteria and archaea. And it has, regions that are very, very highly conserved, which is why we can find it. And then regions that are very variable, which is where those differences come from. And more so, there's a conversation about including more genes in that analysis, though, right? Because we have these sequencing technologies that are becoming more efficient and cheaper and just better in general. And so the conversation moving forward is, do we include more genes in that analysis of how we delineate different taxa? And that's sort of where we are now and moving forward in it.
Thank you. Thank you so much.
You're welcome. Great question.
Do we have any other questions in the audience? Okay, if not, I would say Dr. Gushgari has been very generous with her time already. We've come right up on the hour, so I appreciate you taking the call up from Twitter and putting something together for us from afar. It was really fascinating, and the idea of needing to basically reconsider how we're classifying things when we've been using essentially such a small amount of information to make those major decisions. It really makes you sort of rethink that whole field. So it's fascinating.
Yeah, it's a scary proposition.
Yes.
Well, yeah, thank you everybody for your attention and for the invitation, and take care.
Thank you.